Nowadays, many diseases are treatable with drugs. This drug, for example, has been used to successfully treat the patient. However, before it could be used to treat the patient, it had to go through extensive testing. And nowadays, most testing are still performed in animals before a drug is translated to being tested and applied to human patients. The problem is that many diseases are still insufficiently treatable and we need to develop new drugs. Animal tests can be important in this process, however, they also place the animals under stress and potentially pain.
Our research shows that only 1 in 20 therapies tested on animals are eventually used to treat diseases in humans, that the remaining 19 drugs drop out during this testing process. This 1 in 20 figures show that the process of developing drugs from animals to humans is very challenging. If we could better understand this process, we could potentially optimize the development of drugs. This will not only lead to better human treatment, but also to less animal testing.
Let's assume there are 2 drugs. One of these drugs has been successfully tested in animals and eventually obtains regulatory approval to treat a human patient. And the other drug, despite some promising early findings in the animal testing, drops out in the human testing later because there is a lack of efficacy. What is the difference between these 2 drugs, with one drug obtaining the regulatory approval to treat the patient and the other drug failing and dropping out? Drug development is a very complex process and there will always be failures. However, there might also be parameters in the drug testing which are associated whether a drug works in humans or whether it doesn't. Such factors could, for example, be the trial design under which a drug is tested, or the animal species a drug has been tested in, or which outcomes are measured. Or even parameters we don't even know about are relevant for this process.
Parameters of drug testing, like which animal species has been used, are reported in scientific papers. If we gathered and analyzed all of these parameters for all drugs ever tested, we might identify patterns which could predict successful development of drugs. However, looking at a few scientific papers would likely not be helpful. We would need to summarize many, potentially millions of papers to see patterns. It would be virtually impossible to do this manually. Thus, we at the Stride Lab, we use systematic review, statistical, and data science methods to systematically summarize the data from all scientific papers available to gain a deeper understanding of this process. Once we have gathered all these data in one place, we might be able to see patterns why certain drugs obtain regulatory approval and others failed.
For example, we have shown in a previous research study that if you test a drug under more diverse experimental conditions, for example, testing it in different species or with different model diseases, that increases the odds that a certain drug is later used to treat patients. So if we could understand this process of drug development better, this would allow us to enhance the drug development for both animals and humans. And this could reduce animal testing, but it could also replace and refine animal testing. And on a longer run, it would also contribute to developing and finding new therapies to treat human diseases at a faster pace. We at the Stride Lab are very motivated to make a relevant contribution to drug development for both the patients, but also for the animals and to improve animal welfare.